First of all, I would like to thank the organizing committee for the opportunity to present our data here. Recent progress in understanding the genetics mechanism of lifespan regulation genes provide us opportunity to select the pharmacological touches to influence the lifespan. And the purpose of our investigation was to study the anti-aging effects of some molecules with known effects in aging and longevity. The PI3K and TOR signaling mechanism is the key regulators of cell fates that upregulate the normal cell growth, but downregulate the stress resistance and thus they may be involved in aging process and longevity control. The known inhibitors of PI3K and TOR is the wortmannin and LY294002, and the inhibitor of TOR is rapamycin. The PI3K and TOR signaling pathways are highly conserved, and mutation in PI3K and TOR genes may influence lifespan in so different organisms as C. elegans and yeast and flies. Here the publications where studied the effects of pharmacological inhibition of this signaling pathway, and the effect on lifespan was determined in so different organisms as C. elegans, Drosophila, and yeast. We applied the treatment by inhibitors using yeast paste, and we feed Drosophila wild type strain during all adulthood, during all imago lifespan. The treatment by LY294002 led to increase of median and maximum lifespan in males and females. The effect of wortmannin was observed in males in median lifespan and maximum lifespan, but only in medium lifespan in females. The rapamycin induced increasing in the median and maximum lifespan as in males as in females. According to the evolution theory of aging, the lifespan increasing may have a cost of reproduction, but we don't find decrease in females' fertility after feeding by LY294002. Another important pathway that may modulate lifespan is the NFkB dependent pathway. NFkB is a transcription factor that may regulate genes involved in gene expression, inflammation, and free radical generation. The genes that depends from NFkB increase their expression during aging. So the NFkB pathway is involved in cell senescence and organismal aging. We treated Drosophila imago by PDTS in concentration of 120 macromoles during all lifespan. We find very significant increasing of median and maximum lifespan in males and in females. Also, it was surprising for us. We find that PDTS treatment induced the increasing of females' fertility. That doesn't mean that lifespan increasing after PDTS treatment don't have a cost of reproduction. Also, we studied the locomotor activity that demonstrate the health span of Drosophila. And we found that in the males, the locomotor activity was not lower than before treatment by PDTS. The same data was observed in females. One of the target genes of NFkB is Jumanji. It's a histone demethylase that may control involved in the control of expression genes involved in aging. We found that treatment by inhibitor of NFkB induce the decrease of rate of expression of Jumanji in males and in females. Conclusion. Thus, we have shown that specific pharmacological inhibition of PI3K and TOR kinase, as well as transcription factor NFkB, prolongs Drosophila lifespan. And lifespan increasing is not associated with decreasing in locomotor activity and rate of reproduction. Also, we found that inhibition of NFkB leads to decrease of Jumanji level expression. I would like to thank my colleagues from the Syktyvka and from our laboratory is in the Insttute of Biology of Komi Scientific Center. Thank you.
Okay, thanks.
Mikhail, I have a very provocative question. What do you think about some possibility that you have two effects, specific effects, inhibition of some specific pathways, one pathways, other pathways, and unspecific effects, lifespan prolongation by aromatic effects. And it can be different effects.
Yes, but now we can separate the hermetic effects of compounds and specific effects. But in the case of NFkB transcription factor, we demonstrate that target gene expression decrease. That means that specific effect of inhibitor is play the role. Yes, but we can't separate one from another. I don't know how to say this.
And this inhibition of NFkB, it has to be complete inhibition to get good effects? But only small inhibition?
Unfortunately, we don't study the activity of protein. And it's not possible to answer your question.
But that's
Because maybe in future experiments we will study the migration of NFkB from cytoplasm to nucleus, and then we may say about its activity.
because hormesis experiments show that actually stimulation, little bit 10-20% stimulation of NFkB will be useful. And so that's always to be kept in mind. We often knock out or kill the system completely and then hope for something happening.
Very balancing system.
Yeah. So we are all aware of the explanation at the balance level, but we need to use that idea while doing experiments also. When we do experiments, often we go to the extreme, total knockout, total this thing, which actually does not tell us the real information. So stimulation of NFkB can be good also, and inhibition can also be good.
But we have only experimental results.
No, this is for your future when you
It's just a speculation about it.
Yes. This is for the future, that when you teach your students, then at least theory you can teach. Practical can still be the same thing, we cannot do much. Yes, any other question, comment? Other speaker is here? She has arrived? Ah, okay, we had no, do you want to then use your 15 minutes? Have you given the thing? Okay. Well, Marina Kovina is here now, so okay, so because at 12:45 I will, if I disappear, please don't mind.